Hi, my name is Richard Lin, and I'm a researcher here in Professor Ukhanti Mercy's group in the Harvard-MIT Division of Health Sciences and Technology. The project I'm working on is to quantify and capture CD4 positive and CD3 positive T lymphocytes in patient blood to be used in resource-limited settings. So right in front of me here, we have a microfluidic device that's composed of PDMS, which is polydimethylsiloxane. The device, the channel itself, the height is 50 micrometers high, and the total volume in the device is 10 microliter. So this is an extremely precise device that we're going to use to capture T helper lymphocytes, basically the diagnostic benchmark for HIV patients. So the surface of the microfluidic device has already been prepared with protein and CD4 positive antibodies. In the syringe right now, we have just PBS, biological buffer, at pH of 7.4. And we will now flow this into the channel to flush out the extra CD4 positive antibody. At this stage, the rate does not need to be controlled because we're just trying to flush out the extra stuff. And it doesn't really matter what our rate is. This is where the inlet of the device is going to be, and this is going to be the outlet to my left and to your right here. So now I will flush out the extra antibody solution. And if you can notice from the outlet, you're gonna have the fluid coming out. You know, this is a very minute amount, but if you look closely, you can see that there is fluid coming out of the channel through the tubing. So now that we've flushed out the extra antibody solution, we can now prepare ourselves to flow in, inject in the blood. So I will now unplug the tubing from the inlet. So now we'll open a new syringe for the blood. It's human blood, whole blood. So it's not a patient sample, it's healthy subject sample. Well, we'll prepare ourselves to inject in the blood into the microfluidic device. So we will attach the syringe onto the syringe pumper. Now we'll turn it on, and you can see the flow rate of the syringe, and we've set it to be 5 microliter per minute. And we will now basically prepare ourselves to inject in the blood. We will place the syringe pumper vertically because, as we know, the blood cells do sediment, and we will want the cells to flow into the channel, so we'd rather have the cell sediment to the bottom, which is right near our tubing. So now everything is set up, ready to go. The volume of my device is 10 microliter, and the volume that we want to flow into the channel wall is also 10 microliter. So I can tell if I'm flowing enough blood by just looking at the color of the channel. If it's all red, it's all filled up, then I know I'm flowing 10 microliter, and I should stop the syringe pumper. So now I'll press the start button, and it'll start. So sometimes it takes a while before the blood actually gets to go into the channel, and a lot of it is due to like the pressure. For example, when we put the syringe on there, sometimes we would withdraw or inject the fluid accordingly to adjust the blood in the tubing. So sometimes it may take a while, but once it starts pumping into the channel, the flow rate is set at 5 microliter per minute, which is what we want. So we may have to be a little patient here and wait. Sometimes you have to wait for like 2 minutes, 3 minutes before the blood starts going into channel. Actually, you can see right now the blood is starting to go in the channel. So you can tell right now the device almost completely filled up. Okay, so now we will switch the tubing back to PBS to flush out the blood that's in the channel. And to do so, we will repeat what we already did to remove the tubing. Since we still need to control the rate of the flowing of the PBS, we still need a syringe pumper. However, because we're not flowing blood anymore, we don't need to leave the syringe pumper in a vertical position because there's no cells in the PBS. So now we're all set. And we will now flush out the blood from the device. And I will press the start button again, right here. So as you can see, the device is kind of starting to clear out. The edges is still a little bit of blood, but it is slowly disappearing. So once all the blood is gone, we will increase the rate from 5 microliter per minute to 20 microliter per minute. And the reason for this change is because in the blood, there are actually 2 cell types that have CD4 positive antigens. One is what we were looking for, the T helper lymphocytes. The other one is what we call the monocytes. We want to make sure that our device specifically captures just the T helper lymphocytes rather than both of the T helper lymphocytes and the monocytes. And the method that we do that is to increase the flow rate because we know that monocytes are larger in size than T helper cells. So by increasing the flow rates, the larger monocytes will experience more pressure to leave the surface than the CD4 positive T helper cells. So now it's going to run for 20 minutes. So in my hand right now is a solution. It's called FACS lysine solution. The purpose for this is to lyse red blood cells. So wherever red blood cells are remain in the channel, this solution will lyse it. This is diluted 10 times from the stock solution. So now what we're going to do is stop the syringe pumper, remove the tubing, and basically replace the solution with the one from this syringe. So I'll now stop the machine, remove the tubing, take this off. Take it with the syringe. Replace it with a new one. Okay, so now that everything's set up, we'll decrease the rate back to 5 microliter per minute. So we'll click the rate. From 20, we'll decrease that to 5. And now we'll flow in the FACS lysine solution for 20 minutes, and we'll now press start. So in front of us right now is an optical image of the device taken at 10x magnification. So you can tell all these white dots here are cells. So how can we make sure that these cells are the ones that we're looking for? So we use a couple of different stains to make sure. The first stain we use is what we call DAPI. DAPI stains the nucleus. So basically any cells with a nucleus, such as white blood cells, would all stain. While say the red blood cells, which don't have nucleus, would not be stained. And you can tell that it's all across the image. They're all these blue dots, and every single one with blue dot is a cell. So now, the next step is to check if the cells have what we call the CD4 positive antigen. This is extremely important because the whole purpose for our device is to capture the CD4 positive cells. So the fluorescent antibody we use, of course, is CD4 specific. And we'll go to the next slide. So in this picture, the cells shine out much dimmer is because the fluorescent antibody we use is usually for FACS, so the dilution is much higher. But if you look fairly closely, you can still tell that there are still dots that litter around this field of view. They are fairly dim, but upon closer examination, we can tell that these cells are indeed CD4 positive, which is good because that's exactly what we want. And the next test is, like I mentioned before, there are two different types of CD4 positive cells. One are the ones that we want, the T helper cells, and the other are what we call the monocytes. So we again needed another fluorescent antibody to differentiate between the two cells. So we know that in T helper lymphocytes, they have what we call CD3 positive antigens on them, while for monocytes, they do not. So the next antibody we use is CD3 specific, which you will see in this picture. So again, you can tell that there are dots litter around the device. The dots look bigger because sometimes some cells just shine out brighter, some don't as well. I mean, a lot of stuff are up to chance in this case. But in most cases, these dots, these bright red dots, are all cells. So, which is, again, what we want because we want to make sure that our cells are CD4 positive and CD3 positive. So, in this case, you can tell a lot of our cells are indeed CD3 positive, which is good. It gives us specificity in our capture. The big dots, they could be junk, but a lot of times what happens is sometimes a cell just happens to gather more CD3, fluorescent CD3 onto themselves. So in this picture, this is one of the data that we've collected for this microfluidic device, and it's basically a cell capture program. So in the previous slides, whatnot, you can tell that there were a lot of pictures with cells on them, but what do they really accumulate to? Well, they accumulate to a cell capture profile, in which case we can plot in the x-axis we have the distance from the inlet, and the y-axis we have the capture cell distance. And you can tell that throughout the device, your density initially is extremely high, and then slowly decreases, which is what we would expect because we're capturing the CD4 positive and CD3 positive cells from the bloodstream, and there will be decreasing concentration of those cells in the blood sample. And this is basically how our data is usually presented.